Okay, today I'm going to discuss the concept of mutations that can occur anywhere from DNA to RNA and some of the effects that that might have on a particular protein. What I've written here in this sequence is obviously messenger RNA because you see U's instead of T. But the first kind of mutation I'm going to discuss is what's called a point mutation. And I also like to call this a substitution. I think the word substitution is easier to remember when we talk about what this actually does. If I were to take one of these base pairs that came from the DNA, and again, this is a messenger RNA that came from the DNA, if I were to take one of these bases from one of these nucleotides and actually replace it with another base, we need to look at what might happen. So with this, I'll pretend like that with the glycine here, I'll pretend like that this G was actually changed to a C. So when I do that, when I change that to a CGC, you'll notice that glycine, and on the chart here, that CGC actually now becomes arginine. If it becomes arginine, that is going to totally change the order of amino acids that line up to make this protein. That right there is what is called a point mutation and also called a substitution. If you'll notice, it doesn't affect any of the other amino acids except for this one here. So I'm going to go ahead and I'm going to go back and change this back to a G the way I had it. The other kind of DNA mutation you need to be aware of is what we call a frame shift. Now when I draw frame shift mutations in class, I always draw whenever I draw messenger RNA strands for you, I always put frames around the sets of 3 nitrogen bases. If you recall, those are called codons. Well, what if I were to find some way to actually change the way that those codons are being framed? And so I'm going to call this a frame shift. There's 2 ways I can go about doing that. One of those is called an insertion. The other one is called a deletion. So I'm going to go ahead and erase these frames here that I drew on my strand. Imagine if a mutation occurred and another base was inserted, we'll say right here. If I do that, that has no effect whatsoever on this leucine here. The CUC still stays the same. However, every single set of 3 after that is now affected. And that's going to totally change what that protein would be and probably not make anything worthwhile at all. That is what is called an insertion. I just inserted a base into that sequence. And again, that's a type of frame shift. The other type of mutation that causes a frame shift is a deletion. Imagine if I took out this C and just left it out. I now regroup everything, CUA, UCG, GCU, and so on and so on. And that, of course, is going to completely mess up what protein you're trying to get as well.